Welcome aboard the Bio Train. In this video, we're going to talk about cloning. Cloning is when you take genetic information from one living thing and make identical copies of it. The copied material is called a clone. Geneticists have cloned cells, tissues, genes, and entire animals. Cloning happens all the time in the natural world. In fact, normal cell division, mitosis, is a process of cloning cells. In mitosis, all of the genetic material of one cell is copied before the cell divides into two daughter cells. Each daughter cell has a complete copy of the genetic material from the original cell. The two daughter cells are genetically identical to each other and the original cell, making them clones of the original cell. The process of asexual reproduction also produces clones. To quickly review asexual reproduction, it involves one individual. The new individuals are produced through mitosis, and the offspring are genetically identical to the one parent. Species that reproduce using asexual reproduction have the ability to clone themselves. Cloning occurs naturally in many species, but it also can be done artificially in a laboratory. Most people associate the concept of cloning with cloning in a laboratory, where you're taking a species that would normally not reproduce asexually and producing offspring that are genetically identical to one parent. Scientists thought that it would not be possible to clone complex multicellular organisms like mammals until in 1997, Scottish scientists successfully cloned a sheep. Something that was thought to be impossible was suddenly possible. Let's take a closer look at how this was done. The process is technically called somatic cell nuclear transfer. Somatic cells are all body cells except for the reproductive cells. Somatic cells contain a full set of DNA for that individual. So somatic cell nuclear transfer is when you take the nucleus from a somatic cell and you put it into an egg cell that has had its nucleus removed. Let's take a look at the steps used to produce Dolly. A body cell was taken from a donor animal. An egg cell was taken from a different donor animal. The nucleus was removed from the egg cell. The body cell and egg cell were fused by electric shock. The fused cells began to divide to become an embryo. The embryo was then implanted into the uterus of another foster mother, and then the embryo developed into a cloned animal. The process sounds complex, but it's not as complex as it seems. Let's take a closer look at this diagram to help us understand the process. A donor cell is taken from the udder of the sheep that is to be cloned. This means that all the genetic information that ends up in the clone will be coming from this one sheep. An egg cell is then taken from another donor sheep, and the nucleus of that egg cell is removed. The two cells are then fused. The original donor cell that has a nucleus with a complete set of DNA from one sheep, and the egg cell which had its nucleus removed. The cells are fused using an electric shock. Now you have an egg cell that has a full set of DNA, just like a fertilized egg would have a full set of DNA, except in this case, all of the DNA in the egg has come from one individual rather than half coming from the mother and half coming from the father. The egg cell with the fused nucleus is fooled into thinking that it's been fertilized and begins to divide normally into an embryo. The dividing embryo is then placed into the uterus of a foster mother, and develops into a baby lamb. The baby lamb is genetically identical to the one donor sheep and is a clone of that sheep. Let's review the steps of the process. See if you can understand why this process results in a clone. A body cell is taken from a donor animal. An egg cell is taken from another donor animal. The nucleus is removed from the egg cell. The body cell and egg cell are fused by electric shock. The fused cell begins dividing, becoming an embryo. The embryo is implanted into the uterus of a foster mother, and the embryo develops into a cloned animal. Cloning is now heavily used in agriculture for both animal and plant species to produce genetically identical offspring of individuals with desirable traits. Let's review what cloning is one last time. Cloning is the process of taking genetic information from one living thing and creating identical copies of it. The copied material is called a clone. Geneticists have cloned cells, tissues, genes, and entire animals. I hope that helps you stay comfortably seated on the biotrain.